Dear class 10, we'll continue chapter number 8, that is circulatory system. So this chapter may take some more sessions because the chapter is too long. So today in this session we'll talk about the third type of blood cell called platelets. Okay, and also the clotting of blood. All right. So third type of blood cell that we have is platelets. Platelets, or it is also called thrombocytes. It is also called thrombocytes. All right, thrombocytes. Platelets or thrombocytes. All right. So these platelets or thrombocytes are another important blood cell. Okay, which are oval or round in shape. Okay, non-nucleated. Non-nucleated means they also do not contain nucleus. Okay, non-nucleated. Okay, and the number, okay, its number, that means the number of platelets actually ranges between 2 lakhs to 4 lakhs per cubic millimeter of blood. That means 2 lakhs, 1 tens, 100,000, 10,000 lakh to 4 lakhs, 1 tens, 100,000, 10,000 lakhs. 1 tens, 100,000, 10,000 lakh per cubic millimeter of blood. Okay, the number of platelets ranges between 2 lakhs to 4 lakhs per cubic millimeter of blood. All right, and they are formed from the giant cells. Okay, the giant cells called megakaryocytes. Megakaryocytes. Okay, they are formed from the giant cells called megakaryocytes in the red blood cells. Okay, then its lifespan, the lifespan of platelets, is about 3 to 5 days only. 3 to 5 days only. That means platelets survive for 3 to 5 days only. All right, and they are destroyed in the spleen. They are destroyed in the spleen. Okay, spleen is a part of lymphatic system, so there it gets destroyed. Okay, and your platelets initiate the blood clotting. That means the main function of these platelets is to initiate or help in the clotting of blood. Okay, in the clotting of blood. Clotting of blood is also called coagulation of blood. Okay, slight correction to be made with regard to the formation of platelets. It has been that your platelets are formed from the megakaryocytes in the red blood cells, but it's not red blood cells, but it is red bone marrow. Okay, please do correction there, which means your platelets are formed from the giant cells called megakaryocytes in the red bone marrow. Okay, not in red blood cells, but it is red bone marrow. All right, please correct it. Let us go to this clotting of blood now. Okay, coagulation of blood. Okay, so when a part of the body is suppose injured, okay, or is cut, then what happens is that so there the tissue cells get damaged. Okay, in the injured region the tissue cells get damaged, or your platelets get damaged there. These platelets which are talking about get damaged. So when they are damaged, then they release or they produce a kind of enzyme which is called thrombokinase. Okay, thrombokinase is a kind of enzyme produced by the damaged or injured tissue cells or platelets. All right, and this thrombokinase is also known as thromboplastin. Or recently it is known as factor X also. Okay, it is also known as factor X. Okay, so this thrombokinase, being an enzyme, converts inactive prothrombin. Okay, converts inactive prothrombin with the help of calcium ions into active thrombin. See here are some names of the chemicals. It could be new names for you, so that you need to go through it. You need to just keep in your mind Alright, so you need to keep yourself in touch with these new terms out here. Alright, so this thrombokinase being an enzyme, converts this prothrombin with the help of these calcium ions into thrombin. So what is the difference between prothrombin and thrombin out here is that they are the chemical substance which actually is inactive in this form. That is, prothrombin is an inactive chemical substance found in blood plasma. Okay, with the help of these calcium ions, this prothrombin will be converted into the active one. That is, that active form of this prothrombin is known as thrombin. Okay, thrombin now. See, then your thrombin again convert this fibrinogen. Okay, convert this fibrinogen with the help of this calcium ions again. Okay, that means thrombin with the help of calcium ion converts this fibrinogen into fibrin. Okay, thrombin with the help of calcium ion converts fibrinogen which is soluble in plasma, soluble substance found in blood plasma, with the help of calcium ion, gets converted into fibrin. That means thrombin converts this fibrinogen into fibrin with the help of these calcium ions. And what's the difference between these two? This is soluble, it is in the soluble form, but it is in the insoluble form, which is not soluble. So now this fibrin at the end, forms a network of sticky thread. It forms a network of sticky thread. This fibrin, actually forms a net-like structures there in the injured region. Suppose this part is injured. Okay, so there, after all these things taking place there, what happens is that your fibrin will be formed there. This fibrin makes a kind of net-like structures there. It makes a net-like structures there, which traps RBC. Okay, mainly it traps RBC or other blood cells, and that time your watery pale liquid substance oozes out. It comes out. Okay, it comes out. Only watery pale fluid substance oozes out. So that pale watery substance which contains no fibrinogen, is called serum here. It's called serum. As serum. What is serum? We discussed this in class 9 itself. Serum, about serum. Serum is actually a plasma, blood plasma containing no fibrinogen. That means plasma minus fibrinogen. This plasma, when has no fibrinogen, then that form of the plasma is called serum. It's called what? Serum. Same thing happens out here. This fibrin now makes a kind of net-like structures there, net-like a net or thread-like structures there, which traps all the cells there, and from that trap only watery fluid, pale watery fluid substance oozes out. Okay, which contains no fibrinogen. So that watery substance which has been oozed out from that injured region is called serum, though it is not mentioned in your book, but that is called serum. Okay, then that now net-like structure forms a network of sticky thread, okay, along with the blood vessels, especially RBC, then it gets clotted. It's just what now? Clotted. That in the injured region, a clot is formed which blocks the flow of blood. That means from that injured region, the blood was flowing out. Now, so there a solid, a net-like structure is formed there, which blocks there in the injured region and blocks the flow of blood. So this is how the blood clotting takes place there, and this clot of the blood stops the flowing of blood. Suppose if that clotting doesn't take place there, if no clotting there, if the blood is not coagulated, or let us say if there is no these platelets, then what will happen is that that clot doesn't take place, and from a small injured part of the body also, the blood keeps flowing out, and at the end, the body will not be having the enough blood, and consequently a person may be dying all because of that injured small injury also. So this clotting is very important, where this platelets play a very important role in its coagulation. That's why the blood clotting is very important phenomenon in the injured region. So this is how the clotting of blood takes place, which is also called coagulation. All right. Okay, now we'll see blood transfusion and blood groups. Okay. Blood transfusion and blood groups. From this, what we understand is very much clear to you that when a person is injured and because of the injury, the lot of blood has been lost, or maybe there has been a surgical operation, okay, and the blood has been lost. So in that case, the blood transfusion is needed. That means the blood needs to be given from some other persons, okay, from some other persons, and this practice of giving the blood to the person who is needing, okay, cannot be randomly done. Okay, cannot be randomly done, because if it is randomly done and the compatibility of the blood, suppose, is not there, the blood is not matched, then what may happen is that the reaction takes place there in the person who is receiving the blood, and it may lead to the death of the person also. Okay, before the idea of blood groups, before we had the human beings had the idea of blood groups, people used to randomly transfuse the blood. Okay, they used to donate the blood to the sick persons randomly, and it used to lead a lot of casualties that time. They did not know the reason why it all happened, but afterwards they knew that it's all because of incompatibility of the blood. Alright, so now this blood transfusion is done only after matching the blood, only after testing the blood. If the blood group is matched, okay, if the blood group of the person who is needing and the blood group of the person who is giving, okay, if this blood is matched, then only the blood is donated or the blood transfusion is done. Otherwise, it is not possible, because what may happen is that after the blood transfusion, if it is not matching, the reaction may be taking place there in the person who has just received it, and it may even lead to the death of the person. So it is not done randomly. Okay, so now blood groups, you see, blood groups, that means grouping of the blood. So to blood transfusion, or following the blood transfusion, what is needed? Then now it is very much important that the blood grouping. Okay, so we need to know the blood groups. Okay, if the blood groups are matched, then only it is possible. So what is blood grouping then? So how can we do the blood grouping? So blood grouping is now mainly done. There are two methods. In fact, there are two systems of the blood groups. There are ABO system and RS. There are many, but most commonly ABO system and RS systems are used. Okay, these are the two methods most commonly followed nowadays for the blood grouping. Okay, so this blood grouping, the concept of blood grouping was first discovered by Carl Landsteiner. Okay, so Carl Landsteiner is the person who first discovered the idea of blood grouping. Okay, so blood is composed of different kinds of cells and plasma. So there are proteins on the surface of RBC. Okay, there are proteins on the surface of RBC, and these proteins are named as antigens. Antigens. Okay, it is on the surface of RBC. Okay, there are proteins on the surface of RBC which are called antigens, and these antigens are of two types. They are antigen A and antigen B. Okay, antigen A and antigen B. And there is another substance which is called antibody again. Okay, there is another substance called antibody. Okay, that is in antibody, which substance in plasma, blood plasma. Okay, so in blood plasma there is another substance again in complementary to this. They are antibody, and this antibody is again of two types, that is antibody A and antibody B. Okay, antibody A and antibody B. Okay, so now based on these two types of antigens, antigen A and antigen B, alright, so blood groups have been grouped into four types. Blood groups have been grouped into four types. Okay, let me write here the four types of blood groups based on these types of proteins, that is antigen A and antigen B. Alright, so they are, let me write it here in the book like this: blood group, alright, then antigen present on RBC, then antibody present in plasma. So based on this, there are four types of blood groups. They are blood group A. Blood group A. Alright, so blood group A contains antigen A. Okay, antigen A. And in blood group A, or the person having the blood group A has antigen A on the surface of RBC and antibody B in plasma. Okay, antibody B in plasma. See here. See, blood group A contains antigen A and antibody B. So antigen A is present on the surface of RBC and antibody is present in plasma. Alright, then blood group B. In blood group B you find antigen B, right, and antibody A. See now, antibody A. Alright, similarly blood group AB. Blood group AB will have both antigen A and antigen B, both. Alright, and whereas no antibodies. There is no antibody. There is neither antibody A nor antibody B. Alright, then blood group O. Blood group O, no antigens. No antigens, whereas both antibodies are present. Both antibodies are present. Both antibodies means anti-A and anti-B. See, this is your blood group. Okay, blood group. So how is it classified? how is the blood, or blood is classified according to ABO system. So in the ABO system, blood is classified like this here, as you see here. And as I've told you, there in the blood, we know is made up of RBC, blood cells and plasma. So out of 3 different kinds of blood cells, your RBC contains a kind of protein which is called antigen. Okay, and this antigen is of 2 types, that is antigen A and antigen B. Alright, and in the plasma of blood, there is what? Another substance, that is antibody. Alright, so that way, in presence or absence of this antigens in the blood, it is of 4 types as given here. According to ABO system, okay, there are 4 types of blood groups. Okay, there are blood groups A, B, AB and O. Okay, so according to your ABO system, there are many, but most commonly ABO and RS systems are there or are in use. So according to ABO system, there are 4 different types of blood groups. There are blood group A, blood group B, blood group AB and blood group O. Okay, and so as I've told you, for blood transfusion, compatibility of the blood group has to be seen. Okay, the blood group has to be matched. Okay, so it's very much obvious that blood group A can give to blood group A only, or blood group B can give to blood group B. This is the O to O, AB to AB. But beside this, there is another system also. There is another probability of the transfusion of the blood also there. Okay, so that is, let me tell you in this way. Blood group O can be given to this, and okay, blood group AB. Okay, see now. So this system, this diagram which I have drawn out here, tells you that blood group A can donate the blood group B also, can donate the blood to blood group B also, and blood group O can also donate the blood to A and blood group AB also. That means blood group O, a person having blood group O can donate the blood to the persons having blood group A, blood group B, blood group AB. Okay, that means to all kinds of blood groups, blood group O can be donated. Blood group O can donate the blood. So this blood group O is called universal donor. Universal donor. Okay, blood group O is called universal donor because blood group O can donate the blood to all types of blood groups. Blood group A, blood group B, blood group AB also. Alright? And blood group AB can receive the blood from all other groups. It cannot donate to others. It can donate to AB only, but it can receive the blood from all other groups. So blood group AB is called universal recipient. Universal recipient. Okay, there is other way of donating the blood or transfusion of the blood. So this is the way by which we can make out that O can donate to all other blood groups A, B and AB. So blood group O is called universal donor, whereas AB cannot donate to all, it can donate to AB only, but can receive from all other groups of the blood. So blood group AB is called universal recipient. The summary of ABO blood group matching. Okay, so I have already drawn this chart where you can see which particular blood group can give to which particular group. Okay, which particular blood group can donate the blood to which particular group, blood group other side. Okay, so that can be seen out here. This is the summary of ABO blood group matching. Alright, see here. This blood group of donor. So this side the donors are there, and this side the blood group of recipient. These are the this side the persons who are needing the blood, who are receiving the blood. Alright, now see, blood group of donor A, B, AB and O. Here A, blood group A, blood group B, blood group AB and blood group O of the recipient side. Okay, now see, blood group A can give blood to A. Okay, blood group A. Okay, blood group A can donate blood to A, but cannot donate to B. Can donate to AB. Okay, cannot donate to O. Okay, blood group A can donate the blood to A, cannot donate to B, but can donate to AB and cannot donate to O. Similarly, blood group B cannot donate the blood to A, but can donate the blood to B because of the same type. The compatibility of blood group is there, and can donate blood to AB also, but cannot donate to O. Similarly, AB cannot donate to A. Okay, cannot donate to B also. Can donate to AB because AB can be matched, and cannot donate to O also. Okay, AB can donate to AB only, not to others. Alright, but O can donate to all. O can donate to all. O can donate to all. O can donate to A also. O can donate to B also, AB also, and O to A. That's very much obvious. So the same which I have told you here. So here O can donate to all. From this also you can make out that O can donate to all. Similarly here you see O can donate to all. So O is universal donor. O is what? Universal donor. And similarly AB can receive from all. Though AB cannot donate to all. AB can donate to AB only, but it can receive from all. See here. So it is universal recipient. You can say universal recipient. AB is universal recipient. Alright, this is the summary of ABO blood group matching. We'll see RS system. Okay, RS system. Means RS system means there is another substance there in the blood which is called RS factor. This RS stands for rhesus. Okay, rhesus, which means a common monkey in whose blood this factor was first discovered. Okay, so it is RS factor. Alright, and most of the people are RS factor. That means they have got, most of the people are RS positive, means they have got RS factor. This factor they have got. But there are people who may not have this RS factor also. So they are RS negative. Okay, they are what? They are RS negative. That means those who do not have this factor in their blood, so they are RS negatives. But most of the people are RS positive. That means they have got RS factor. Okay, so now during the time of blood transfusion, suppose blood is given to a person who is RS negative. Suppose, okay, blood is being given to the person who is RS negative by the person who is RS positive. Okay, RS positive. Suppose he is donating the blood to him. who is RS negative. Okay, so in the first blood transfusion, it will be okay. There is no any problem. Okay, the normal blood transfusion will be there. But after almost about after about 2 weeks, okay, after about 2 weeks of the blood transfusion, this person develop a kind of antibodies in his blood. Okay, antibodies will develop in his blood after about 2 weeks of the blood transfusion. Okay, and in the second blood transfusion, again, if the second dose of the blood transfusion is given, okay, then what will happen? This antibodies will sensitize, okay, or this antibodies will react, which means there will be the reaction in the blood of this recipient, which may even cause death, which may even cause death of this person. All right, so that it should be matched. Not only that the group, but even this RS factor needs to be matched. So that problem might be caused. Okay, then now another, this is RS factor in pregnancy. Okay, or in pregnancy. RS factor in pregnancy. Okay, see, suppose there is a RS negative woman. Okay, suppose RS negative woman. There is an RS negative woman who is pregnant, and the baby which is developing in her is suppose RS positive. Suppose RS positive, because father may be RS positive. Okay, because father may be RS positive, because of that the baby may be RS positive, but this RS positive baby is developing in the RS negative woman. So in that case, what happens is that the first baby will be normally given birth. There won't be any problem in the first delivery. But in the second case, because once it is formed, then suppose this this RS positive is conceived. In the first case, is not the baby will be normally given birth, but in the meantime, in this woman, that your antibodies will be pumped. Okay, then in the second case, immediately suppose the second, the lady is again, or the woman is conceived second time immediately, then what will happen is that there will be some complexities. Okay, there will be some problem in the growth or the development of the baby inside. Okay, so that complexity may be leading death of the fetus there, or it may lead to abortion of the fetus there. Okay, so that's because of the unmatched RS factor there. So that during the blood transfusion, and this factor has to be seen very seriously, and sometimes this problem may even take place because of this unmatched RS factor. Okay, so today we'll do only this much, and we'll continue in the next session. Thank you very much.